Got some delicious food, some dim sim. So my name's Cam, and I'm the chair for this afternoon's session. Thrown in the deep end. I wasn't expecting on doing this, so we'll see how I go. Fingers crossed. Bear with me. So the first speaker we have up is Christine Houghton. She spent the past 30 years working as a nutritional biochemist and is the managing director of Cell Logic, an Aussie company specialising in nutrigenomics solutions for enhancing health. I was just having a chat with her before I came up, and she's a bit of an advocate for nutrigenomics, surprisingly enough. Who would have thought? She says it's completely changed the way that she thinks about food, and she's about to do the same for you. So sit back and enjoy. Welcome, Christine.
Well, thank you, Cameron, for that kind introduction. Yes, actually, after 30 years in practice, I would have to say that knowing what I know now about nutrigenomics has completely changed the way I would practice nutritional medicine. So I'm going to try and share some of that with you this afternoon. And first of all, we have to start with the dietary guidelines. Population health obviously has to put out guidelines which sort out recommendations that suit most of the people most of the time. The one-size-fits-all model. But you might be the one that this doesn't fit, and this is where nutrigenomics can step in to help. So what we try to do with nutrigenomics as a science is to look for aspects which are clinically relevant, and we try to look for ways in which we can change our clinical practice in keeping with what the genome tells us. Interestingly, nutrigenomics is a new and evolving science, and it's quite a complex science in that most of us have either come in from one side or the other. So I've come in from the nutrition biochemistry side, other people come in from the genomics side, and what that means is people like me who have a handle on the nutritional biochemistry have got to somehow get a very fast handle on the genomics side of things. But what nutrigenomics is doing is really driving the paradigm shift in the practice of nutritional medicine, and it's giving an added dimension to the term personalised medicine. So what do I mean by reshaping the nutrition landscape? Well, what I mean is this. If we start with a model where we've looked at dietary guidelines as a foundation, the one size fits almost all, we're looking now for a more inclusive model. So you as a clinician can tailor your program more to suit the needs of your individual patient. So as a nutritionist, I'm interested in what's in food. Food isn't just vitamins and minerals, proteins, fats, carbohydrates. There are a whole host of phytochemicals and other compounds in food that we have in the past and in keeping with the nutritional guidelines have mostly neglected. We want to see how we can take those food bioactives to alter the phenotype of the patient. And in between all that, there are now a whole host of omics sciences which help us to get to the end game. So what do we mean now by recommended dietary intake? It kind of changes everything. And this paper from 2011, published by Professor Michael Fennick, who works out of CSIRO in Adelaide, is one of the movers and shakers in the world of nutrigenomics, and he has considered this idea that the way we've looked at recommended dietary intakes now needs to change. And he says that nutrigenetics and nutrigenomics hold much promise for providing better nutritional advice to the public in general. And so his paper here gives an overview of various aspects of nutrigenetics, which is the testing side of the genome, looking at the various SNPs a patient might have, and the nutrigenomics, which is the part relating to the nutritional recommendations that one might give. So how these are relevant to practitioners and to their patients. I have to point out what nutrigenomics is not. There is now a whole movement, and any quick trip to Google will show you that there are a lot of people who think that methylation is all that there is to know about nutrigenomics. Methylation and the MTHFR polymorphisms are not what we mean by an all-encompassing definition of nutrigenomics. I want to give you an example of some of the things which appear on websites, forums, where consumers write in and they express their anguish because they're coming up with poor advice that Dr. Google is giving them. And here's an example of an exact commentary. So maybe someone can offer some help. I've recently been diagnosed with C677T homozygous, as if it's a diagnosis and a disease. So I was taking this much B12 and this much folate and da-da-da, was on it for 2 months, felt tired, heavy, jittery, went off all the supplements for a week and was told I should try 1/5 of a tablet of B12 for 1 week, and if I don't feel well, to stop and go on to 1,000 milligrams of 5-methylfolate. Then it leaves you kind of hanging what to do next. So I was then advised, if you don't feel well taking half a capsule, stop taking it. And you're either receiving sufficient methylfolate at this time or you're not yet prepared to take it due to other mutations present or other health issues worsened by methylation. So the patient says, okay, which is it? I'm receiving enough, I have other mutations, or what? Very confusing. And so it goes. So that's the sort of nonsense that's going out there in Google land, where people believe that this MTHFR gene and its polymorphisms are responsible for practically everything going wrong with them. Why am I so concerned about megadosing with 5-methylfolate? Well, I'll tell you. 5-methylfolate reductase, the MTHFR gene here is responsible for producing the 5-methylfolate. If you have polymorphisms in the gene here, which means you will produce an enzyme which isn't as efficient as it could be, you will be limited in the amount of 5-methylfolate you can produce. Therefore, if you take a 5-methylfolate supplement, you are bypassing the fact that this enzyme isn't working properly. So therefore, if the RDI for folate is about 400 micrograms a day, and you're taking a supplement, why would you need to take more than 400 micrograms a day? So what is actually happening is supplements like this, to be bought online, this is 15 milligrams, 15,000 micrograms of 5-methylfolate. So you think it's only a vitamin, it's expensive urine. No, it is not. It is actually causing some severe adverse effects, and the likely result is severe oxidative assault. So what that means is, if we're going to interpret a nutrigenetic profile, we can't just look at one gene in isolation or a handful of genes. We need an integrated approach. And I'm more interested in looking at a whole host of genes which are associated with the way human cells support their own defence systems. And so I teach a model where, in order to look at cell defences, we're looking at a whole host of factors here. So we're going to look at redox balance, we're going to look at inflammation, genes associated with energy production, genes associated with detoxification, and in addition to that, methylation. But each of those, in isolation, tells you very little. We really need to look at them all combined together. So, are we ready yet is often the criticism that's put to clinicians working in the nutrigenomics space. Pure science would say, no, we're not ready yet, we have insufficient evidence, we really need more information. And so you will see lots of papers like this looking at whether or not we're ready. Regardless of whether we can get a direct answer to that or not, my answer is this. We have consumers online getting very poor advice. Whether we think we're ready or not as trained clinicians, I think we owe it to the community to become sufficiently informed that we can at least steer people away from bad choices. So we have entered the era of personalised nutrition. Here's a 2013 conference which was looking at translating nutrition, integrating the research, the practice, and trying to develop some policy guidelines. So what came out of this conference was this, that there are significant challenges in translating SNP data into personalised advice, no question. Many SNP diet studies don't show a cause-effect association, and not all of these SNPs are validated, and I think that's a very important point to note, because there are companies putting out huge numbers of SNPs on their reports, but a lot of those SNPs are not validated. In other words, you can't translate them into a significant clinical outcome. Classical genetics is a different story. It's associating genotypes with specific disease risk. We're not looking at the risk of specific diseases. We're looking at altered upstream biochemical processes that cells use to support themselves. So if we can look at a patient's nutrigenetic profile and see that perhaps the genes which code for the phase 2 detoxification enzymes, for example, are not likely to be operating at their peak, then we can start to piece together some of the likely scenarios which may or may not be related to a particular disease. So in personalised nutrition, the genetic data relate to the optimal diet for a given genotype with the goal of reducing disease risk. And of course, I think consumers would welcome such personalised advice. So it's an evolving era. We don't have all the answers, but we certainly have a lot of the underlying science already to be able to give good advice. So this paper from Keith Grimaldi, published in 2014, looked at a number of fairly commonly known and understood genes and showed that these are scientifically validated. So things like MTHFR, superoxide dismutase, phase 1, CYP1A2, and some of the glutathione transferase genes. That's not all that are validated, but that's some of the better known. So we look at different gene-diet interactions in complex disease and looking at that in terms of public health policy. So it makes it more difficult on a public health basis, but still we have information which we can use to make recommendations which perhaps change the way we look at recommended dietary intakes. As an example of what one might see on a nutrigenetic profile, here's an example of a case where this individual has a normal superoxide dismutase gene, that's a mitochondrial SOD, but has abnormal glutathione peroxidase and catalase. Now, to put that into perspective, what that means is this. So superoxide dismutase is the gene which takes the primary superoxide radical and combines that to produce hydrogen peroxide, which is itself a reactive species. So that's what SOD does, but now to complete the process, to completely quench that radical effect of the superoxide, we now need glutathione peroxidase, which takes the hydrogen peroxide, turns it into water and oxygen. So in this patient, the MN-SOD is working effectively, but these two genes are SNPs, which are coding for variant enzymes, and as a consequence of that, this individual may need to look at redox pathways with some concern. MN-SOD, the manganese superoxide dismutase genotypes, have been associated with risks of diseases like breast cancer and prostate cancer and a whole host of conditions. I think our most important consideration in any of this is rule number 1, do no harm. So how do we translate this information from the lab bench to the clinic? So you start out with your nutrigenetic report. You have to understand some of the nutritional biochemistry, and this is the area that I'm focused on teaching. If you don't understand the biochemistry underpinning the gene, then it's very hard to interpret what to expect. So the nutrigenetic test just tells you what you were born with. It doesn't tell you how your body's expressing that gene. So you need to look at some test pairing. So you might want to do some blood tests or urine tests or other tests which can enable you to understand how that patient is actually expressing that gene. They may have changed their lifestyle in such a way that that gene is being expressed optimally. So we need to know that. Sometimes we have to consider the epigenetic phenomena. We can then look from an intervention point of view at culinary genomics. What food is going to upregulate genes as we may need it, and then we may need to give nutrigenomic prescriptions on top of that. And that's what we mean by translational nutrigenomics. So what are we actually trying to achieve here? Well, what we are trying to do is we're trying to look at food as a source of compounds that give a direct effect and those which give an indirect effect. So, for example, if we have a trace element like selenium, that's a nutrient giving a direct effect. So the glutathione peroxidase enzyme doesn't work without selenium. So if the patient has inadequate selenium intake, glutathione peroxidase won't function normally. The indirect effect then is a phytochemical like sulforaphane, which acts as a signalling molecule and switches on particular genes in that way. And then we have metabolic effects. So a lot of polyphenols, like the polyphenols that are found in olive oil and green tea and red wine and so on, have to be metabolized, and those metabolites then send signals to the cell. So we have a whole host of ways in which we can affect that genome. If we go back to superoxide dismutase again, the different forms of superoxide dismutase require copper, zinc, manganese, depending on what form, and then as we saw, the glutathione peroxidase requires selenium and catalase requires iron. So they're direct effects. And then we look at indirect effects like a whole host of phytochemicals which are activating the cellular switch, NRF2, and the antioxidant response element. And so what they are doing is they are now upregulating a whole host of different genes. NRF2 is a most fascinating compound because many researchers now are showing it's the key to cancer prevention. And so you see papers like this that say there is strong evidence that increased expression of the antioxidant response element regulated genes inhibits cancer development. So that's why I'm so interested in using compounds that affect and upregulate NRF2 as a means not only of protecting against cancer, but of protecting against all chronic disease. Here's another paper from 2004 that showed that if you can upregulate the NRF2 pathway, you're going to switch on a whole host of antioxidant and detoxification genes. So this is the key to getting to the cell's own defence system. If you have a look at the pictures on the left here, you'll see the healthy cell and the way in which it takes care of itself, and on the right, what the cancer cell is doing. So on the left, the healthy cell naturally upregulates its phase 2 detox enzymes, but the cancer cell downregulates them. On the cytochrome P450, this is the phase 1 enzymes. Healthy cells downregulate them, cancer cells upregulate them. So understanding the biochemistry underpinning these genes is very important for our ability to interpret. There's just an example of a number of the NRF2 target genes and the processes which are involved. So again, just reinforcing the importance of the biochemistry and a number of the effects. So if we look at one example of these processes interacting, that's the Krebs cycle. The first rate-limiting enzyme in the Krebs cycle is an enzyme called a aconitase, and if you block aconitase, your ATP production is reduced. What blocks aconitase? Superoxide. So if you have a redox imbalance whereby superoxide is excessive, you will block your ATP production. Okay, we'll skip through that because I know I'm running right out of time and have probably already run out. Let me just give you quickly a case example here. We're just going to look at estrogen metabolism. So the prime goal of the detoxification pathway in estrogen metabolism is to prevent the formation of DNA adducts, which are mutagenic. So there's estradiol going through a number of steps. So estradiol presented to the cell is acted on by the phase 1 enzymes, That now produces a 4 or a 2-hydroxyestrogen. COMT, or catechol-O-methyltransferase, now methylates that estrogen and makes it inactive, and then it disappears by glucuronidation. We then, if that continues along the path towards DNA adduct formation, we now have glutathione S-transferase, which is a phase 2 enzyme, which now combines glutathione to conjugate that compound and get rid of it out of the system. The key point here is we can still, at this point, have these toxic quinones, mutagenic quinones, about to attack the DNA, and there's one further step that the cell uses. It has a very potent enzyme called quinone reductase, which it then takes this quinone, cycles it back to the beginning, and round the loop it goes until finally we have eliminated the DNA adduct. And at that point, I have run out of time.